Hi, everyone. Welcome to our weekly meeting on the cerebellum. Today we have Mireille Broucke. Mireille is a control theorist who happens to be very interested in applying some of the ideas in control theory to understanding the cerebellum. She received her electrical engineering degree at University of Texas, Austin. Then she went to University of California, Berkeley, where she received a master's and a PhD. And she stayed another year, did a postdoc there, all the time working on vehicle dynamics and autonomous robots, multi-robot coordination. She then became a professor at the University of Toronto, where she has stayed her entire career. In 2017, she published her first paper on psychophysics and learning associated with sensory motor systems and humans. And since then, she's been interested in applying control theory to neural computations that take place in the cerebellum. And in particular, the idea that the cerebellum trains the reflexes and the importance of forgetting in the training process to add robustness for control. Recently, she had a review and annual reviews of control where she talks about some of these details and introduces and encourages basically control theorists to think about the cerebellum. I'm so happy to have us learn from you today.
Thank you. Thank you for the invitation. Okay, so indeed, I will be talking about the internal model principle, and I will start right with my focus is on the ocular motor system, so we will get right into it. And I have here most or part of the neural circuit that's associated with the slow eye movement systems, which I'm interested to explore from a control theory point of view. And the particular focus then is going to be on these three eye movement systems, the VOR, gaze holding, and smooth pursuit. So I know that this group is an expert on the saccadic system, so that's not going to be covered, although you'll see some of the circuitry for the saccadic system here. So my aim is to model the flocculus and the ventral paraflocculus, which together are called the floccular complex. And to do that, to start sort of that work, I have to very carefully determine what is the open loop model. The open loop model is the model in total darkness, the dynamic behavior that you get in total darkness, which includes various parts of the brainstem and, of course, the oculomotor plant. And the other thing I have to do very carefully is figure out what are the measurements. And of course, we know that there's a head velocity measurement coming from the semicircular canals. But I also have to sort of map out what kind of measurements are coming from the visual cortex and other places in the brain. And so I should say, as a control theorist, it's quite difficult for a control theorist to dig into the anatomy, to read all these papers, and to figure out what's going on. So there can be inaccuracies, and if you see something that doesn't sound right, please let me know, because I'm certainly not an expert on this neural circuit. But what seems to be happening is that there are two types of visual measurements. One is a retinal slip velocity measurement, which seems to be coming from the knot and the MT. And these are used for the optokinetic reflex and also for the nodulus uvula complex, which is not presented here. And so that's one distinct measurement. And there is, it is interesting to note that there is a projection of this retinal slip velocity down to the NPH in the brainstem. The other grouping of regions here are related to a positional measurement, a visual error measurement, which I'll call the retinal error, which is the distance from a target to the fovea on the retina. And this error measurement seems to be generated by, of course, the FEF is generating this measurement for the saccadic system, but there's a dedicated region for the slow eye movement system, which was a discovery of various people, such as Krauslis and others. So there is a dedicated region of the FEF for the slow eye movement systems, and there's also a similar region in the rostral superior colliculus that gets this measurement. And these seem to be sort of parallel systems to form this measurement. So this is the measurement that will be used in my modeling work. I'm not directly using the retinal slip velocity here, although there are indications that it could play a role. In particular, if you lesion these regions, you see an effect on smooth pursuit, but it's an effect that recovers after about a week. So it's a little bit mysterious what this other retinal slip velocity is doing besides this projection here. I'll be doing modeling work based exclusively on this positional target error, which I call retinal error. Other points I would like to make are that there's interesting behavior going on in here. You have MT, which is generating signals that are visually contingent. You must have, for example, a target displayed for there to be responses in cells here. But this region, MST, mysteriously has extraretinal signals. And there's been quite a lot of study of these extraretinal signals. And the consensus, I can tell you now, that seems to be reached by a number of people with various papers about this by Ono, Mustari, Lisberger, and others, is that there is a corollary discharge from the output, the Purkinje cell output of the floccular complex back to MST. So this is interesting because this is a new wrinkle that internal models are operating in parallel, and you can have I would call a primary internal model for the slow eye movement systems in the floccular complex, but perhaps some kind of secondary behavior going on in this other location, which is projecting to this not very well understood dorsal paraflocculus. So here I have 4 modules of the cerebellum. And as I said, I'll be concerned with these 2. I'm interested in modeling these 2. And these have been included here simply to show you some other parts, but to say that all of these regions have somehow been implicated in the slow eye movement systems. Finally, I want to mention that there's also been a lot of research on the frontal eye fields, particularly a kind of gain control mechanism. That becomes interesting and relevant as we start doing our modeling. Okay, so, and of course, there's a great deal to be said about the medial vestibular nucleus, the abducens nucleus. This NPH has a neural integrator. We have to spend a lot of time, in fact, modeling that part, because if you don't model that part properly, you won't get the right predictions about the floccular complex. Okay, so that's our setup. That's we're kind of mostly concerned with the middle strip there, but we've put some of the extra parts on the side to let you know some things that we are not going to be modeling. So starting from a control theory point of view, we start with an open loop model. So we have to start with total darkness, and we have no regulation from the cerebellum, and we have no visual signals. Well, I actually have a visual signal here, so that's not completely true. But anyway, we start with an oculomotor plant model, which is the standard model going back to the 1960s with David Robinson. You can make a second order model if you want, if you're interested in acceleration terms. This is a sort of velocity-based model that is adequate for the slow eye movement systems, and it's sort of a standard off-the-shelf model. And then we have here this retinal error that I mentioned. This is all going to be about horizontal eye movements. I'm not dealing at all. And by the way, the neural circuit I showed was horizontal eye movements. I'm not talking at all about vertical eye movements. So R is the position of a target in a world frame. X is the angular position of the eye with respect to the head, and XH is the angular position of the head in a world frame. If we take our neural circuit and we flip it over on its side, we get sort of what looks like a standard so-called unity feedback loop in control. So if you open up a third-year control text, you'll see a picture like this. You won't usually see a box at the top. It'll be somewhere embedded in here, but no worries. And this is a kind of picture that Ito would make. He would talk about sort of the cerebellum as a side loop, and that if you remove the cerebellum, you still have sort of a basic functioning primitive loop through the brainstem. So that's a little bit what's conveyed in this picture by placing the cerebellum at the top and not in this main path. And this represents the plant. So this is a, very simplistic, high-level description of the previous neural circuit that we saw. And then we begin modeling the brainstem, which is modeling the NPH and the MVN. And here it immediately becomes complicated because there's actually a lot going on in there. And I've here simplified a little bit some of the steps that are needed to get this model of the NPH and the MVN. This is basically the so-called brainstem neural integrator that resides in the NPH. And there's a little bit under the hood here. There are some adaptation processes going on to adapt these parameters A hat and B hat, which are supposed to be adapted to match the A and B up here. And these are adaptation processes that are not mediated by the cerebellum. They are mediated by that knot projection. And that's been shown in experiments with goldfish, in fact. So this is a very primitive process that doesn't tie to the cerebellum. And what happens is that if A hat adapts to match A and B hat adapts to match B, then this is an observer. This is basically a Lundberger observer of the oculomotor plant. It's an adaptive observer. And what it does is it generates X hat, which is a signal that gives you eye position. And that can then be distributed throughout the brain to be used by any other brain region that needs that information. These are the reflexes. So this is the VOR. This is the head velocity, which we get from the semicircular canals. And this parameter here is called the VOR gain, which has been studied extensively. There is another reflex that's really not usually, I don't think it's usually called a reflex, but it behaves so similarly that we've grouped it with the reflex component of this motor command. And it has a gain as well. And that gain is important for the second time constant of gaze holding. So there's 3 time constants of gaze holding. There's a time constant that comes from the oculomotor plant. There's a second time constant that comes from the NPH plus the oculomotor plant, and that is coming through this signal. And then the third time constant comes from the top up from the cerebellum. So then we form a motor command from what we have available, and we have not yet, we're still doing the open loop model. We have not yet modeled what comes out of the cerebellum, but we're already labeling it as an internal model because of what's going to follow. And so there are 3 components here, and this is meaningful for both the discussions in the oculomotor literature as well as the discussions in the visual motor literature. So let's start with the visual motor literature that you all know very well. The visual motor literature talks about 2-rate models. So there's a fast system and a slow system. What that corresponds to here is a fast component and a slow component. The fast component is responsible for sort of the transient part, and the slow component is responsible for the steady state part. You have the same kind of discussion going on in the oculomotor literature, where they talk about 2 systems driving the slow eye movement systems. You see that in the literature, people talk about a system that's open loop. What they're actually talking about there is the first 100 milliseconds, where visual signals and cerebellar output are being formed, and these are being driven by sort of very fast, signals that come in 15 milliseconds, 50 milliseconds, to generate a fast response to initiate smooth pursuit, for example, or initiation of smooth pursuit is often preceded by a saccade. So if we imagine, a scenario where there's no saccade, so using one of these paradigms where you don't get the saccade, then this would be a purely smooth response. It's also interesting to note that, the way this is supposed to work with these 2 signals is that initially this will be a large signal because it will be driven by visual errors. And this will not have developed at all because the cerebellum needs time to build this signal. And then as time goes on, there's sort of a redistribution of energy, and this signal goes very small and this signal goes large. And that's precisely the picture you will often see in the visual motor literature of these types of sort of like shifting of magnitudes of signals. So in the control literature, we don't use, slow, fast types of terminology, but we say this is responsible for the transient response, and this is responsible for the steady state response. Okay, so the next thing that we have to do is now we have identified the error signal that we're interested in. This is the error signal that would be regulated to 0. We have an open loop model, and we need to know, from a control theory point of view, we need to know what's happening to that error. If that error is to be regulated to 0, so that basically all the objectives of the slow eye movement systems amount to driving that error to 0, then what is happening to that error? So we make a kind of virtual model. We make an error model that tells us the evolution of that error signal subject to the dynamics of the open loop model and the signals that are already pre-specified, like these reflexes. And what we find is that we have an error model whose, first of all, stability properties are determined by this term. And it turns out that this reflex, which remember is responsible for the second time constant of gaze holding, is actually destabilizing this error model. So we have, first of all, a critical issue that we have possibly unstable behavior of this error. We have a visual signal that we can use to do something useful. We have another signal coming out of the cerebellum that we can do something useful. And we have a piece that's very important. This is an effective disturbance. It is a mix of target velocities, target position, and head velocity, scrambled up in a sort of way that's because of the filtering through the plant and because of these reflexes. And this signal is not measurable directly. This is not available to the brain. Now, there are parts of it that are measurable through the reflexes, and I'll get to that later, but essentially taken as a whole, this is not measurable. So we have to drive an error to 0 with a disturbance acting on these aerodynamics that's not measurable. And this is precisely the setup of the internal model principle. Okay, so basically 2 things have to happen. If the brain is going to be able to manage this situation, the brain will have to stabilize these dynamics by using this visual signal. And this signal here has to cancel all these terms. Okay, so now as a control theorist, we see this, and then we say, well, what's going on in the oculomotor literature? Is there any evidence that this is really happening? And what you see is that over a span of, since the 1960s, what people have effectively been doing is running experiments that probe this signal. So you have smooth pursuit experiments where R dot is non-zero, but the head is held stationary, so these terms are 0. You have gaze holding experiments where this term is 0 and this term is 0, but this term is not 0. You have VOR experiments where this term is 0, this term is 0, but this term is not 0. Then you have experiments of adapted VOR gains where you see phase shifts in the response that's observed, and that's due to sign changes. There, you can even change the sign of this term here. You can similarly do experiments that adapt this parameter, but this parameter seems to be a little bit less well understood, so I have not actually seen directly the experiments that would correlate with this parameter. So we look in the literature and we say, okay, so what do people say? And of course, we have behavioral experiments that were really the immediate attraction when coming up with the idea of looking at the internal model principles. It was the behavioral experiments that drew me to the area. But if you really want the proof, you have to look at these experiments on the horizontal gaze velocity Purkinje cells, which have primarily been, you can see a lot of these papers come from Lisberger's group. And these papers are recording directly on this class of Purkinje cells. And what you see in each and every case is you see the signal above. You see that what's coming out is this signal. So this signal is being reproduced on the Purkinje cell output of the floccular complex. So that is what I'm taking as the proof that there's an internal model in the floccular complex. It's that information that I get from these papers through these various paradigms. There's also, of course, studies that study perturbations. And so we also want to match the transient response in the various papers that study that. But I'm not taking that as proof necessarily. I'm really taking this as a kind of proof. So our claim then is that basically for the floccular complex to generate these signals at the Purkinje cell output, even though it cannot directly measure those signals, that is an instantiation of the internal model principle, which basically says every good controller must contain an internal model of all reference and disturbance signals impinging on a control loop. And this is basically saying that in steady state, those signals have to be built. You cannot expect to achieve perfect tracking if you don't build those signals in your control loop. That was discovered in 1975. So there's two papers on that, 1975, and this is the 76 paper. So this is the discovery of Bruce Francis and Murray Wonham. Okay, so now we have a hypothesis. And at this point, we take our open loop model and we augment it with a visual signal that can stabilize that error model. And this is just from the control literature on how to do that. So we're not informed at this point by any considerations of the neural circuit. It just will be a windfall that it will happen to correlate with the neural circuit. That was purely by chance. So we happened to take a design from some Italian colleagues, Sarani, Marconi, Isidori. They have probably the simplest internal model design that you can use in this context. So it's an adaptive internal model. And there are other internal models. And if you're wondering, well, how do I pick one? It's going to depend on how you arrange the mossy fiber inputs. That is the determining factor in which internal model you need. So this one is a very simple one. It's basically a stable filter. It's taking the ocular motor command here bundled together, but in fact, there are different mossy fiber inputs. So these mossy fiber inputs actually have to come in on different channels, but here it's been all bundled together. And this is the aggregate behavior of all the parallel fibers, which have to synchronize. So this is a stable matrix. So this makes this a stable filter. So we basically have filtering going on, which is presumed to go on in the granular layer. And we have an adaptation process, which is the synaptic plasticity at the parallel fiber Purkinje cell synapse. It is driven by the error signal, which is the error from the inferior olive on the climbing fiber input. This is a gain, which is simply setting the speed of adaptation. And this in adaptive control, this is called the regressor. So the regressor in this is the parallel fiber signals that are coming in from the granular layer. And then due to the, with the adaptation of this parameter is then weighting. It's taking a linear combination of these parallel fiber inputs and forming the output on the Purkinje cells. And it's very simple. I mean, it's remarkably simple. And the question is, what is it doing? Here's what it's doing. This is a stable filter. This F is stable. So initially, imagine that initially there's nothing happening. So this term is absent. So initially the cerebellum is simply in a sort of a quiescent state with nothing happening, and then a signal comes in. This signal includes all these pieces. In particular, it includes this piece. This piece comes through a sort of recurrent loop, in here, and it's added in here, and you see that what happens is this F will be replaced by F plus G psi hat transpose. Now, as the psi hat starts building up and starts adapting, what it's doing is it's taking the poles of this stable F, which are in the open left half plane because it's a stable filter, and it starts dragging those poles over onto the j omega axis in order to generate persistent signals. Because what the cerebellum is having to do is deal with persistent excitation coming in, persistent signals that need to be canceled. And this is, of course, a very dangerous operation. So very, very dangerous to start to insert this marginally stable internal model of these persistent signals, sinusoids, sums of sinusoids, steps, ramps, stuff like that. So that is what is going on with this parameter adaptation. It is trying to build a model that reproduces the frequency content of those disturbance signals. Okay. Okay, so now some principles that come out of this. If this is really going to be the explanation of the floccular complex, you can only have an internal model satisfying the internal model principle if you take the Purkinje cell output and send it back to the mossy fiber input. And there is such a pathway verified in a number of papers. There is such a pathway. But more generally, there is something called the nucleocortical pathway, which has been verified for many species and for many cerebellar modules. And that would be an ingredient, that would be a candidate for this sort of closing the loop, if you want, from the Purkinje cell output to the mossy fiber input. And this is something that, of course, has been hypothesized by others on purely experimental grounds. Lisberger has written a lot about this. But it comes up again when we look at how the internal model principle needs to operate. It does need that pathway, or you don't have the behavior corresponding to the internal model principle. So gain control. So theoretically, the feedback gain that's here, that needs to be large enough as a function of disturbance parameters. Those are the theorems that we get from our Italian colleagues. So it is interesting that, in fact, the frontal eye fields do supply a kind of adaptive mechanism. So my Italian colleagues did not speak about adaptation. They simply said the gain has to be sufficiently large. But in the brain, of course, the brain doesn't know what are the disturbance parameters. It cannot anticipate what they would be. And so it's really very interesting that the brain has a mechanism as smooth pursuit progresses to look at the magnitude of the internal model output and use that to adjust this parameter, to basically increase this parameter. And this is entirely what would be predicted from a purely theoretical side. So that's an appealing windfall of having chosen this internal model design that I chose. Safety. Okay, safety is a big deal. I think perhaps now people are studying this a little bit more, but I think originally in the cerebellar literature, this was not mentioned so much. So inserting an internal model in the cerebellum is a very dangerous proposition, because there you have a model that's going to generate persistent signals like sinusoids and ramps. Now, if you remove the error signal, that thing will keep going. It runs on its own. Remember that the fast system starts with a large signal that's error driven and it drops to 0. And then the slow system ramps up. That's the internal model output, and it just keeps going with 0 error. And this is, of course, verified in error clamp experiments and experiments where you occlude the target. It's clear that the cerebellar output continues. So now you're going to need to carefully manage that. And one way that the brain could do this is by the following mantra, that for correct operation of an internal model, when the internal model output goes high, the error should go low. And if not, you're going to have a race condition. You're going to have high errors with high internal model output, and you will force that internal model to go unstable. Because remember that the poles of that matrix, F plus G psi transpose, are on the J omega axis, and a slight tweak pushes them to the right, and you get unstable behavior. So the brain, for it to manage this whole setup, which is a remarkable thing, it's going to have a suppression mechanism that when it detects the cerebellum is running too hot, cool it off. And there seems to be such a thing, and this is the nucleo-olivary pathway, where you can have the Purkinje cell output suppress the inferior olive activity. And there's another, as I was, writing down this talk, I was actually thinking there's probably another, and this is speculation, but there's probably another safety mechanism going on, which is this projection from the NPH to the inferior olive, which would say when the internal model output goes high, then X hat, the eye position, should go low. You don't want tracking at very eccentric positions of eye angle, because you're going to push the eye beyond its range. So you'd like to have some head movement, something to help. So this might be another protection mechanism, another safety mechanism, but that's pure speculation. There's an issue that's beyond really the scope of this talk, but it's also, I think, not completely explored in the cerebellar literature, is that for correct parameter adaptation, there must be excitation. And you have certain signals coming in on the mossy fiber inputs that are not exciting, in particular this one, which is supposed to be driven to zero. And this informs a little bit how mossy fiber inputs can be organized together. There's also a notion that if you're going to have mossy fiber inputs coming in on different channels, there is a requirement that these stable filters that are receiving these inputs have to synchronize on the same time constants. And this is something that we use in control theory called Kreiselmeyer filters. So this is something we take as for granted because we implement our controllers on computers. But if you're going to actually implement such a infrastructure in the brain, then, well, how is the brain actually going to do this? And so I don't know very much about the granular layer, but I do believe that theoretically speaking, there's a need for some kind of synchronization mechanisms. And then finally, there's another consideration, which is that the cerebellum is doing a major job of adapting reflexes, and this puts architectural constraints on how the modules are organized. And I had thought I was going to sort of get into this a little bit more, and I think I'm going to leave this for another day because it requires sort of building up from what we have at this point, which is simply the floccular complex. And so what I'll do is I'll use this as a segue to get into the next part. And the next part is really the question that, if the cerebellum, in fact, is housing internal models of the type that I'm describing, then how does it come to pass that it's responsible for long-term adaptation of reflexes? What's the relationship like? How is this possible? And here I'm going to be a little bit more control theoretic. This a little bit broadening out. I have to say that I'm presenting this work to control theorists. So I'm also trying very much to appeal to my control colleagues. So in a slightly more general setting now, imagine that you have a single input, linear time invariant error model. So again, this is this abstract model that's characterizing the behavior of some error. So these are now a vector of error states, not just the scalar error that I had before. And I have, again, a disturbance that's sort of the effective disturbance after I do the modeling work to arrive at this error model. So I have some error states that basically I want to drive to zero. I have my usual motor command or input, and I have some disturbances that I want to reject. And the reflex problem is one where there are also sensory measurements coming in from the body. And so here I have a vector of P sensory measurements. In our VOR example, we had only one, but now we are generalizing the setup. And these sensory measurements somehow give us some direct measurements of some parts of this disturbance. So you can think of like the VOR gives us some measurement of head velocity, which appeared in certain terms of that complicated disturbance signal that I had. Now, these sensory measurements are multiplied by some gains that are not known, that have to do with pathways that are not modeled, like the pathway from the semicircular canals to the point at which the disturbance enters this error model, something that perhaps we have not modeled. And there are also disturbances which are simply not measurable by the body. For example, the velocity of a target. The brain has no direct information about that. So there's a splitting of this disturbance between some parts that can somehow be sensed by our sensors and some parts that we just can't get a handle on, and that we can only sense through something like a visual error measurement. And then our setup here, the general setup, is that we'll use the same motor command that we had before with our fast and slow components, our internal model output, our visual signal, and we will add reflexes, which will have a gain that's adaptable. So this gain is now going to be adapted. And this setup is taken straight from, reading the literature on reflex gains. We basically here with this setup, we're trying to mimic what we think is the closest analogy in a sort of a control setting for what seems to be the general concept of reflex gains. So this is a vector of reflex gains. And then the question we have is, how are these reflex gains adapted, and what does it have to do with internal models? Okay, so I'm gonna put out a design that's going to be based on what I've just done. So I have a sort of control problem. This is actually quite weird problem. I want the usual regulation requirement that my error states will go to 0 on a short time scale. And this is the work of the cerebellum, to vanquish errors on a short time scale. I also want any reflexes that are around to sort of work more than the cerebellum. I want to prioritize, like if I'm just doing a VOR type behavior, I want that reflex to handle that. I don't want my cerebellum there working to take care of sinusoidal disturbances. So I have an optimality criterion that for control theorists is not natural. I can tell you this is not existing in the control literature. Prioritize one control over the other. My colleagues say, like, what is that? When we give talks on this, they put us in the advanced control section of the conferences because they say, why would you need that? And then the other requirement of robustness is simply that if I have reflexes that measure directly certain disturbances, then impulsive disturbances that arrive on those measurements should immediately be canceled so that they don't affect things like eye movement. And that's, of course, a very natural requirement. The one that's weird is this optimality thing. So what we do is we take an internal model design that's basically a little bit souped up from the previous design. So we have the previous design that gave an estimate of the signal D. We then do some filtering on our sensory measurements because sensory measurements are noisy and we don't want impulsive disturbances to come in through these through anything on the internal model side. So we filter sensory measurements so impulsive disturbances don't go there. And then we have our standard gradient law, but with sort of an extended regressor going on with this added filtering. So it's sort of a bumped up internal model, a little bit redundant, in fact. We have the same visual signal here or stabilizer, whatever. We have our reflexes and we have our same internal model design. So what's going on? This internal model is redundant and it is reconstructing the D plus the reflexes, which obviously is redundant. But look at what happens. You take the D, which has this formula that we wrote at the beginning, and you add to it this reflex term, and you see that there is an alpha hat minus alpha. So this generates a estimation error on the alpha hat. It's an estimation error, which now our internal model must reconstruct. So what does the internal model do? It reconstructs those disturbances that are simply not measurable, and it reconstructs the residual error due to poorly calibrated reflexes. And this is the key insight to understand why an internal model is responsible to calibrate reflexes, is that if this is quasi-static, then in steady state, due to the internal model principle, the internal model will in steady state cancel this signal because it's driving errors to zero rapidly on a short time scale. Now, the less accurate is alpha hat, the more work must be performed by the internal model due to the presence of this residue. Therefore, we can take as a cost of operation of the internal model to be the steady state cost on the output of the internal model. So we make a quadratic cost and it's in steady state. And now we compute the gradient of that cost. And that gives us a gradient law for reflex adaptation, which we run slowly so that the internal model output has time to go to steady state. And it uses as an error signal the output of the internal model. So the internal model output becomes the new error for the downstream process of reflex adaptation. And the regressor of this new adaptation process is the sensory measurement. And we place the epsilon very small in order to impose a separation of timescales. And now something very nice comes out, because this is the idea of Lisberger. You see, Lisberger wrote this fantastic paper, and also the Miles Lisberger paper of 1981 is an absolutely beautiful work, where they say, you know, there's two sites of plasticity. They have a site of plasticity in the MVN, medial vestibular nucleus, which is running slowly and which is storing the information that's learned from the cerebellum. So you have a transfer of learning from the cerebellum to, in this case, the brainstem, but more generally, it would be from the cerebellum to the deep cerebellar nuclei. So in terms of what this does with our oculomotor system model, basically everything stays the same. I have added some filtering here on the head velocity signal. I've placed it in the brainstem. I don't know if that's right. Maybe that filtering is going on in the cerebellum. There's various arrangements that could be made. I could also include the alpha sub H hat here. The one that's here could be included here. One could make modifications on this, and effectively the behavior is the same from the point of view of the internal model design. The new adaptation law is here, and that would, of course, be sitting in this block B. Yeah, that's how the VOR would look. Okay, I have to speed up a little bit. So I want to mention that what's remarkable about this design is its ability to capture impulsive disturbances and keep them separate from what the internal model is doing. There's a simulation here with a first part here for 10 milliseconds, or I believe it's 10 seconds, where it's a standard VOR. This is the head position, head velocity. The retinal error is rapidly driven to zero, and it's just a standard VOR. Eyes goes opposite to the head. Then with an experiment where the subject is tracking a target that's related to head position, if you make this a parameter here, -1 in the experiment, you need a short-term doubling of the VOR. So now the head moves twice the amplitude of, the eye moves twice the amplitude of the head. That would normally mean that you need a doubling of the VOR gain, but the VOR gain is being adapted slowly. So when we look at adaptation of the VOR gain, it's barely moving because this is only over 20 seconds. Meanwhile, there is an impulsive disturbance that comes in on head velocity because the person with their feet, they hit the ground, and that causes a little jiggle on the eyes. And that is detected by the semicircular canals. And what happens is, because the VOR gain has remained at 1, which is the true value that you need on that pathway from SCC to its arrival in the error model, that needs to be 1 to cancel that. And indeed, it is canceled, because it's canceled instantaneously by the reflex. And you can see that in the retinal slip, which is very small. If you didn't have an architecture like that, if you had an architecture that somehow didn't use 2 timescales, only had 1 parameter, so you have a single timescale design, you would have to double the VOR gain to get short-term regulation of the error. And then you would see large retinal slip due to impulsive disturbances. So this is an ingenious design that uses reflexes to cancel impulsive disturbances, but leaves the cerebellum to do its work to deal with the persistent disturbances. Okay, so I'm going to have to speed up a little bit. I appreciate that you're allowing me to talk a long time. And I'll go to sort of generalizations of all of this. So there is another reflex gain here, right? And I've now explained to you how this one works. It's an energy management scheme. And so I'll be very brief in explaining this one. This reflex is thought to be concerned with gaze holding. It destabilizes the plant so that in total darkness, the eye can hold gaze at an eccentric position for 4 seconds or something like that. And that is the interpretation of the reflex. But remarkably, it really looks to us like this is yet another energy management situation. So this reflex gain, if you follow a theoretical development that's the same as what I just showed you, with writing the error model, having a 2 timescale architecture, what we find is that there's an identical, there's an identical adaptation law for this one as for this one. And so this is a new interpretation of this reflex. It's not just about gaze holding. It's really energy management. The cerebellum is doing energy management. And it's also interesting, and I find exciting, that these ideas actually are new for control theory. This is not just, oh, hooray, control theory is able to say something about the cerebellum. It's actually the cerebellum is saying something to control theory, because control theorists do not have a theory of optimal control for steady state response. We only have control theory from Kalman that regards the transient response. So we are actually, with my colleagues, there's colleagues in power systems and in other areas that are actually working on developing a framework of control, optimal control for steady state response. And it was largely for me driven by the discovery that this is another energy management scheme. And so I have a little bit of, theoretical explanation. There's a cost function. It's the output of the internal model, the output of the cerebellum. One can look at the problem from the point of view of the average cost for the cerebellum or the instantaneous cost. At the end of the day, when we go and make our model, we see that there's the same type of adaptation process that runs slowly. The error signal for that adaptation process is the output of the internal model. And now the regressor is the output of the NPH, that adaptive observer in the brainstem that forms the regressor for this process. These slides, I'm trying to run now, I'm sorry to have too much material, but these slides are really trying to bring home the point of reducing the work of the cerebellum. With reflexes, the cerebellum works very hard. Without reflexes, the cerebellum works very hard. With reflexes, it sometimes does no work. Here, with reflexes, without reflexes, the cerebellum works hard. Sometimes the cerebellum needs to keep working. Depending on the arrangement of disturbances, you sometimes have to have reflexes and cerebellum work together. Sometimes the cerebellum can literally turn off. It does nothing. Concluding remarks. Okay. We have shown that the internal model principle provides a reasonably complete account of behaviors associated with the floccular complex. Long-term adaptation of reflexes can be interpreted as an energy management mechanism to minimize the steady-state operating cost of the cerebellum. For 40 years, the community has embraced the hypothesis that the cerebellum operates based on forward models, yet no one has been able to derive a specific forward model that accurately models the behaviors of, I should say, the floccular complex. I don't know about other modules. And so what I want to put forward as a provocative proposal is that the hypothesis is that the cerebellum is more likely driven by internal models subserving the internal model principle of control theory. Now, some people believe the cerebellum performs diverse functions that cannot be captured by a single principle. That may be, and it is an open question. These are the papers I've been talking about, and thank you for your time and your attention.
Well, Mireille, thank you so much. Wonderful. So let's open it up to questions from Mireille. I'm gonna start first, I guess. I was surprised with the cost function that you had for the cerebellar output. It depended on UIM, but there was no error, which was the purpose of, basically the purpose was to reduce the dynamics, make the error zero. So can you just briefly explain a little bit about why the optimization depends only on basically a quadratic function of U?
Yeah, so it is very interesting, and it's completely new in control also. Control theories have not seen such a architecture. The error that's driving the adaptation in the cerebellum is the true error that we want to drive to zero. So that's what's the climbing fiber input on the climbing fiber input to the floccular complex. And so this gradient law, it could be taken as a law that you would get by writing a quadratic cost on the error, on the actual error. Okay, but then what seems to be happening that's remarkable is that then this signal becomes a new error that's propagated downstream, informing downstream processes to sort of say, look, guys, I'm just working too hard here. And that becomes the error. You see how this sits right in the same slot as this one? So on the short term, these parameters adapt to drive this to zero on a short term. And on a long term, these parameters adapt to drive this error to zero on a long term. And thereby sort of transferring that work, pulling it away so that essentially the cerebellum can lie quiet.
The Purkinje cells, you mean, in this case?
Yes.
Wonderful. Yeah. Other questions for Mireille?
Do you not find that plausible, perhaps?
Yeah, I think that your description is basically saying that their output of the cerebellar cortex needs to be a transient that in the long term goes away.
If it can, but in some tasks. So for example, if you have someone doing fixating on a target straight ahead and doing a VOR, you'll have very little modulation of the Purkinje cells because the reflex is doing all the work. But if you have a task that the head is stationary and then the eyes have to track a sinusoidal target, now the Purkinje cell output is large, and especially if you do VOR cancellation. Now you have a large output because the reflex doesn't give you the proper signals to offload the work of the cerebellum. So it's very task specific.
Hello.
Yes.
Can I ask the question?
Yes.
Yeah, it's extremely beautiful, but like the math is a bit intense here. So, like I know, but like I'm just trying to get an intuition. So how's that? How the math build a form model? Like I think somewhere you show that how you are able to get a new internal model into the cerebellum here, and then replace it when the, like for example, the external environment change. Is that part of the theory?
Yes, yes. And by the way, I mean, the main challenge for the entire effort here is making sure that the math is accessible and believable. And that's, of course, one of the biggest problems I face is that, so you are a representative person who I have to somehow communicate with, yeah, that's not easy. So I want to say that the idea of inserting an internal model. Oh, one thing I should say, I have refrained from using the word exosystem because it's a strange thing to talk about an exosystem. But what it is signals that arrive from the environment, like sinusoids, sums of sinusoids, can be translated into differential equations. And so we associate to signals a system that we call an exosystem. And it is simply a differential equation that generates those signals. So the internal model principle, as it was conceived, was that through the interaction of this process and the adaptation of these parameters and this term here that goes into this equation, that what would happen is that in steady state, this equation would become the exosystem. It would become the differential equation that generates the required signals. And that's a lot to take. That is that you don't just like, oh, I believe that. Believe me, you don't just believe that. When you first see that, like we teach that in undergrad, and people are like, forget it, I don't get that. It's so abstract, really, for anybody the first time you hear it. And then if you hear it and hear it and hear it and hear it, then you're like, oh yeah, sure. So that is the way it's described. It's an exosystem that generates signals, and we are creating that adaptively in the cerebellum.
Adrian, did you have a question?
Yeah. So if I understand the internal models that you're describing they're models of things that are happening in the outside world. So maybe a model of the movement of a target or something.
Yes.
When we talk about internal models in motor control in the cerebellum, often what people have in mind there is a forward model of the plant that is being controlled.
Yeah.
And I was just curious if you could comment on what the relationship is between those two kind of internal models, and whether you believe in internal forward models of your own plants, or do you mostly think the internal model principle tells us about internal models of external exosystems?
So I haven't yet run across a reason to put a forward model into cerebellum. But mind you, I've only looked at the floccular complex and a little bit at the saccadic system. So from this perspective, I haven't felt compelled to do it. And the main reason is that the oculomotor system already has a forward model here. So it's hard to motivate, why would it need another one? The other thing is that if you look at the output of the Purkinje cells, it just doesn't look like a forward model. The VRR cancellation is a great example. The VRR cancellation, the eyes don't move, but the Purkinje cells are strongly modulated. So now the Purkinje cells are doing something that's completely different from what the plant is doing. And so there's just a conflict. You can't resolve that if you are going to say that a forward model is in the floccular complex. For the floccular complex, it's just too hard to make forward progress, no pun intended, if that's going to be the opening move. So it's more propitious to make this opening move and then to see the consequences for reflexes and so forth. Now, going forward, though, looking at other cerebellar modules, I mean, we have to be open-minded. We have to consider all possibilities on the table. But I will say this. The cerebellum seems to be able, at least from the lessons of the floccular complex, seems to be able to behave in different ways according to the experiments that are being run. So if it's VOR cancellation, if it's gaze holding, it seems to be able to deal with all those scenarios. A forward model, instead, is an object that is characterizing something that's an invariant. The fact that I have certain kinematics of the limbs, that's going to be that way for the rest of my life. It doesn't depend on when I'm lifting a cup. Okay, there's something new. I'm lifting something heavy, something unexpected. Yes, the cerebellum wants to know about that. But the cerebellum isn't concerned with the fact that my arm is basically kinematically identical through most of, of course, there are changes. So from a point of view, again, of sort of parsimony, I would like to keep brain regions for forward models where those are structures that remain largely invariant. Whereas the cerebellum is something that's sort of quick reacting. Oh, conditions have changed. I'm going to do different things. To me, that makes more sense. So duplication of effort, forward models here and forward models there, doesn't sound reasonable to me. And also sort of what's part of me that's invariant versus what's coming in from the environment that I need to respond to. I think that's a boundary. That's a meaningful boundary that we want to sort of capture. I don't know if that somehow. So that is my position. If you're asking, what's my position?
Yeah.
I remain open-minded, having not studied.
You are
but I have already a little bit of a worry, a concern about placing something that's an invariant in the cerebellum.
Yeah, okay, thank you.
That was just lovely. What a wonderful discussion there. Are there any other questions for Mireille? Yeah, go ahead.
I would just like to ask, I don't know, a more general question maybe, because this is something that I was also thinking for some time, but in another way. But my question really was, how would this sort of an approach in control theory generalize to, planning and control, other than just, sensory regulation kind of control and like discrete differential equations?
Yes, you asked the million-dollar question. So okay, so there's an elephant in the room here, which is that I started with a linear time invariant error model, very restrictive. Okay, and now what I think you're hinting at is, why don't I have discrete time models? Why don't I have nonlinear models? Mm-hmm. So there is a whole area that is undeveloped in control theory where we need to really generalize our notion of an internal model. Mm-hmm. We don't have astoundingly, even though we've had the internal model principle for 50 years, we don't really have a full theory of internal models for nonlinear plants like the limbs, the legs. Okay, so people are actually working on this, but this is relatively unexplored. For something like the saccadic system, we are way behind as control theorists. We have very simplistic discrete time models to talk about the, disturbances entering into the saccadic system or visual motor adaptation. These models are very, very naive, really miss the true story of how bursts are generated and things like that. So we don't have that. I have colleagues that are working on sort of burst systems and things like this. And one in particular that's interested in the internal model principle with respect to such systems. We have to figure that out. That's something we have to figure out. So nonlinear models, bursting type models, and then the other one, time delays. So how is it that classical conditioning is related to the cerebellum? Well, time delays can be viewed as another form of disturbance. But now we have to be more flexible in what we mean. What's the internal model for a disturbance? So we have to now develop some mathematical tools that would allow us to model time delays. And that would give us an entree to also work on classical conditioning type problems. So I don't know if I fully answered your question, but it is to say that it's a humbling moment for control theory because we don't have any of this stuff. So what you see is the simplest thing we could do. Linear systems, time invariant. And there's a lot to do. And that is why I want my control colleagues to jump in, because we need to get going on this, you know.
Thank you.
Thanks for the question.
Thank you, Mireille. That was just fantastic. It's just so inspiring. It's so clarifying, the ideas that you presented. I'm so grateful that you took the time, and thank you everyone for your questions. I hope you have a wonderful day.
You too. Thank you for the invitation. It was really wonderful.